So once again, hello everyone. Happy World Engineering Day 2021. Today we have the 9th session of the HBP Tea Slides, a mini seminar series that we started almost a year ago, actually, to promote the students within the HBP and their work. Today we have two speakers, Chiara and Antoine. Each of them will have 10 minutes to talk about their research, and then we're going to have another 10 minutes or so, maybe more, depending on how many questions you find attendees ask in the Q&A box. And hopefully we're going to have a lovely discussion. So you can also, instead of typing a question in the Q&A box, you can also raise your hand and ask it verbally if that's what you prefer. So as I was saying, the speakers were kindly volunteered on account of winning the awards for best presentation and poster, respectively, at the HBP Student Conference about a month ago. So I am 100% sure that these presentations will be extremely interesting for everyone attending today. The first presenter is going to be Chiara. She is a PhD student investigating the role of sleep in learning and cognition. And I will hand over the floor to Chiara now.
Okay, so hi everyone, my name is Chiara. I'm a PhD student at La Sapienza University, and today I'm going to tell you about the work we are developing towards the investigation of the role of a deep sleep-like activity on internal representation of memories. So, so to do so, we implemented a simplified two-layer thalamocortical network relying on minimal assumptions. Still, we want these assumptions to be biologically plausible. Specifically, we show that sleep performs a density-based clustering, meaning that similar memories are grouped together during the sleeping phase. This leads to an improvement and a gain in energetic consumption from the network and an optimization in the information carried. Specifically, a density-based clustering has such advantages, and it does not require any a priori information concerning the number of clusters, the shape of these clusters, and it is completely independent from the number of elements involved. Then we suggest two different methods for entropic and energetic measurements in order to evaluate the effect of this density-based clustering on the network structure. We propose two of these methods because we want them to be experimentally reproducible, indeed, because we would like in the future to compare the prevision made from this model with experimental data in order to validate it. Then we show that a deep sleep-like state also improves the network performances on classification of new unseen examples. And all these findings are also reproducible in a more biological spiking neural network. So I'm not going into much details about the network structure. This is a two-layer structure represented in the diagram on your left. And the thalamus projects to the cortical layer and back through a symmetric synaptic matrix. The cortical layer is also provided with recurrent cortico-cortical connections. After the training phase, all the memories will be encoded into the synaptic weight matrices. And specifically, according to the particular training protocol we are adopting, we will have the different subpopulation of cortical neurons will be maximally responding to different examples, so to different memories. I'm not going into detail about this, but during the sleeping phase, we will assume a Nebbian plasticity for the synaptic connections. And we randomly extract a population in the cortical layer performing an upstate. Then we perform some maths in order to evaluate how this upstate affects and modifies the synaptic weights. Here's our results. You can see there are two terms. The first order concerns the thalamocortical connections, whereas there is a second order of attraction due to the presence of recurrent cortico-cortical connections. What I'd like you to note is that in both these contributions, there is this cos theta term. This cos theta represents the distance between two examples. So the more these examples are similar, the closer these examples are in the space, the more the synaptic weights will be modified. In order to give you a qualitative intuition of what is happening, we trained this network on an extremely simplified data set that is depicted in panel A and D. This data set is made of blue and red points that are described by only two features, the X and Y position in the space. So at the beginning, we have this internal representation of memories, as you can see in panel A and D. Then look at the top line for only the effect of the first order of approximation, and in the bottom line to have an idea of what happens if we also take into consideration the recurrent connections. In both these situations, after the sleeping phase, we have the situation depicted in panel B and E. You can see that red points that were closer in the space also at the beginning of the sleeping phase are even more close, and specifically they are also collapsing into one single point in the space. Blue points that were much spread at the beginning are clustering into two different clusters, one on right and one on left. The data set that was not linearly separable at the beginning increases its separability during the sleeping phase, reaching a complete separability in the end. The presence of recurrent cortico-cortical connections makes this process quicker, much faster. Then we wanted to make it a little bit more biological. Thus, we just dropped the assumption that all memories are stored equal. Specifically, some memories are better learned than others. And we evaluated it by associating to each memory a number that we call mass. More massive examples will lead to a higher probability to perform an upstate during the sleeping phase. This means that during the sleeping phase, high mass will behave as a basin of attraction for different clusters. We show here in panel A a situation in which we have a pre-sleep memories with three massive memories depicted by large points. In the end, we will have three clusters forming and no more big points. This is because we also implemented an homeostatic effect of sleep that leads to a regularization of the heterogeneity of these examples. So in the end, almost all examples will be represented in an equal way. Okay, I gave you just a general idea and intuition of what is happening. Now we wanted to perform some more quantitative measurements of these and how this affects the network structure. To do so, we provide two different modalities. The first one is a structure-based measurements of entropy and energetic quantities, and these represents the formal way to perform them. Still, these need a prior knowledge of the synaptic structure of the network that is not experimentally accessible. Thus, we suggest also an alternative set of measurements that are based on the activity of the network that can be experimentally measurables. And we aim to look at these kind of measurements in order to validate our model by comparing these results with the experimental ones. Going into more detail, looking at the green line, you can see that the energy consumptions during the sleeping phase in performing a classification tasks reduces over time. This means that the more we sleep, the less energy we need to perform the same task. This is obviously a gain for the network. Also, looking at the purple line, you can see that entropy reduces during the sleeping phase. This means that we need less amount of information to describe the internal status of the network. Also, we measured the cross entropy, which is depicted by the blue line that increases during sleep. This is telling us that even if we need less amount of information to describe the internal status of the network, we are not losing any kind of information. This is like as if we're just compressing the internal status in a zip file. So we need less amount of information to describe it, but we are not losing anything. This is obviously a gain for the network. So to sum up, we built a simplified two-layer thalamocortical network. We show that sleep naturally performs a density-based clustering that leads to an improvement in energetic consumptions and for information needed to describe the network, so from an entropic point of view. We also provided two different ways to measure these entropic and energetic quantities in order to have a set of measurements to be comparable with experimental results. We also have found out, even if I'm not describing it here, that sleep also leads to an improvement in performances over a classification task, also for harder data sets, such as the MNIST one. And finally, all the findings I have told you here are completely reproducible in spiking neural networks that also are more biologically like and so interesting. So thank you very much for your attention. And if you have any question, I will be happy to answer you. Thank you.
Thank you very much, Chiara, for that lovely presentation. I can't wait. I've already written down a whole page of questions to ask you. And we'll get there after Antoine's presentation in the Q&A session. And that will give our participants just enough time to get their thoughts in order and put the questions in the Q&A box. Wink, wink. Okay, so our next speaker is Antoine. And he has a background in psychology and music cognition. He is also currently a PhD student working on bridging neuroscience, the neuroscience of creativity with digital arts. And I'll now leave it to Antoine to talk to us about pareidolia, if I'm not mistaken.
Yeah, you're pronouncing it well. I will share my screen. So hi everyone, I'm very happy to be here today sharing our recent work entitled Probing the Creative Mind Through Paridalia Induced by Fractal Patterns. So first of all, what is creativity? So creativity has been mostly defined as the ability to produce work that is both novel and appropriate. And it has been widely measured with the divergent thinking task. However, it has been related to tolerance to ambiguity, as well as altered perceptual experiences, characterizing the psychedelic experience and psychotic episodes, to name some examples. So it raises the question, do creative individuals perceive the world differently? Here you can see a quote of Da Vinci talking about how he looked at geological structures and how it fed its creative inspiration. So what he is describing is instances of pareidolia, which defines as the perception of significant objects in noisy or ambiguous stimulation. So one classical example would be to look at clouds, for example, and seeing meaningful shapes such as a cat or a dog. So we decided to study the perceptual components of creativity by developing a pareidolia task. So to manipulate pareidolia in a laboratory, we decided to use images that varies in terms of their fractal dimension. So fractality can be defined mostly as a probability to retrieve similar patterns at different magnitudes or scale. And it has been shown that images with fractal dimension around 1.3 both facilitate pareidolia and are qualified as more aesthetic. So we had three main objectives. The first one was to replicate the effect of fractality on visual pareidolia with the hypothesis that mid-FDs will be associated with increased pareidolia. And second, we wanted to confirm that pareidolia can serve as a perceptual marker of creativity with the hypothesis that creative individual will perceive more objects and more often in ambiguous stimuli. And third, we wanted to investigate the interaction between creativity and stimulus properties in the emergence of pareidolic percepts. So our hypothesis was that fractal dimension and contrast will moderate the relation between creativity and pareidolia. So to measure creativity, we used self-reported measures. We had questionnaires focusing on the phenomenological, personality, and productive aspects of creativity as complementary measures. We expected that the phenomenological measure of creativity will correlate the most with pareidolia, as it might better reflect perceptual components of the creative experience. So we will investigate if that measure of creativity can predict results in a pareidolia task. So our design, first the stimuli were cloud images generated by a fractional Brownian motion algorithm and ranked according to their fractal dimension. We tested 50 neurotypical participants and they had 8 seconds to detect a maximum number of percept in each of the 360 stimuli. So you have example of stimuli on the left side with different fractal dimension and the 3 level of contrast. So we derived 3 dependent variables from that design. So the first one was pareidolia occurrence, which is a binary variable. And then we also add the number of objects for each trials, as well as the reaction time. So first of all, the effect of fractal dimension on pareidolia. We can see here significant quadratic trend illustrated by this inverted U shape here. So we show that low to mid FDs facilitate pareidolia and mid to high FDs are associated with increased number of pareidolic percepts. So this replicates the previous results showing that mid-range fractal dimensions, or around 1.3, facilitate pareidolia. And then our second question, which was related to the relation between creativity and pareidolia. So we found first that the 2 pareidolia measures, which are here, were significantly correlated with the phenomenological measure of creativity. We also show that the 3 measure of creativity were correlated with the number of perceived objects, and as well the creativity, the phenomenological measure of creativity was correlated with the 2 other measures of creativity, which shows that they partly measure the same construct. And here on the left, you can see pretty strong correlation between our creativity measure and the measure of pareidolia. So then we ran mixed effect model to predict pareidolia. What is interesting here is that we see that there is a 3-way interaction between fractal dimension, creativity, and contrast. So we follow up with postdoc analysis. So here is an interesting pattern for high contrast images. So we see that this images with the fractal dimensions around 1.3, like this inverted U shape, is only present for low creative individuals, while high creative experience pareidolia more constantly across all fractal dimension. So this shows the 3-way interaction. So to summarize, we show that fractal dimension around 1.3 do facilitate pareidolia. We also reveal that high creatives experience pareidolia more often and find higher number of objects when it occurs. And also that fractal dimension and contrast are good moderators for distinguishing low and high creatives engaged in pareidolia. So now we can ask what could be the possible mechanisms explaining that creativity pareidolia relation. Creative individuals might use pareidolia as a source of aesthetic experience. So as it had been mentioned, fractal dimension around 1.3 has been associated with aesthetic and perception of beauty. So we might ask the question, is perceiving the beauty in the world fundamental to our capacity of engaging in creative processes? Other possible explanation will be that pareidolia may foster remote associations by connecting initially unrelated concepts emerging from ambiguous stimuli. So we know that chaos and disorder has a function in cognitive perceptual processes. There has been studies showing a relation between brain entropy and divergent thinking, which is classical measure of creativity. And also there is the concept of complexity matching, which shows that in specific perceptual tasks, there is a matching between the entropy of the stimuli and the entropy of the brain activity. We can speculate that entropic or ambiguous stimuli might foster higher higher levels of entropy in the brain through complexity matching, and that higher levels of brain entropy may in turn be associated with increased creativity. These hypotheses may be tested in future work. So we are running now, starting to run a MEG study with similar paradigm. We also think of doing follow-up analysis with multifractal images, and also the idea of linking pareidolia with remote association by including measure of originality of the percepts. So these are ideas that will be tested further on. So this is all for me. Thank you for your attention, and I will be glad to answer any questions.
Thank you very much, Antoine. This is going to be a lot to unpack, I think, for both of you, and I am very excited because we have questions pouring in left, right, and center, and I hope you're ready. So I am going to start off with Chiara, and then I guess potentially alternate between you two. I haven't decided yet. This is an absolutely an ad hoc system. So question for Chiara. Very cool results. Love how you have considered ways to verify experimentally too, all that you've done. Have you considered how hippocampal activity during sleep might fit with your model?
Thank you for the question. Not really. We have tried to implement some measurements that might be easy to perform from an experimental point of view, such as the measurements from the firing rate, so from the activity of cortical neurons. Still, we'd like to, this is a future work. We'd like to talk with experimentalists in order to really understand how we can implement this in an experiment, in a biological experiment, and then to how we can compare these results in a, complete way. Hope
very good.
That answered.
I expect so. Otherwise, they can raise their, anyone can raise their hand at this moment and ask a question live if that's what's required. And you can raise your hand if you feel like your question hasn't been answered fully, and you can offer additional information, additional questions. Oh, actually, I have a question too. You said sorry, Antoine, this is a burning question that I have. You said you can give certain memories a mass in your model. Would you sort of feel confident to say that this is basically like a correlate of an emotional tag to a memory? The more intense an emotional tag to the memory, then the higher mass it has, and thus you can remember it longer? Or is this too much of a stretch?
That's quite too much. I mean, that might be almost anything. It might be that we were, more ready or more interested in that aspect, or we just, we associate it to something that happened in the day. That might be almost everything. We just think that we hypothesize that not all memories are stored equal. We don't remember everything the same way. So we try to model it through the association of this mass number to each memory that in the spiking model is modeled as a different recurrent connections intensity in the cortical layer. So again, a higher probability to perform an upstate.
Right.
Yeah, thank you.
Perfect, thank you. Okay, Antoine, your turn for the grilling. So the images you analyzed are mainly black and white. What about color images? Can you say anything about those? How would, sorry, I need to parse this sentence. How would they, I guess, how would the color images be correlated with pareidolia and creativity?
Okay, that's a good question. The idea, because there is not a lot of work that has been done with this, we wanted to be the more restrictive in our approach as possible to tackle to manipulate stimulus property properly. So this is why we use black and white, and so then we are sure that it's fractal dimension and contrast that have this effect. So we were more thinking of going into multifractal images instead of colors. The idea is that for pareidolic structure to emerge, these are really like Gestalt mechanisms. So you have like figure ground segregation. So for example, with black and white, I think it's easier to have those pop-up of those images that come out of the images. Otherwise, with colors, it will be, I think, a little bit more difficult to understand what is going on, especially if we go with follow-up analysis in MEG or EEG. We want to have really something like a clean paradigm. So color would be maybe a little bit too complicated in the paradigm.
Right, that makes sense completely. I also had a question. I can't remember exactly what it relates to in your talk. So there's obviously, so you're looking at contrast and you're looking at FD, sorry, that was
fractal dimension or fractality.
Yes, so you're looking at the relationship between FD and contrast on the ability of a human, not ability, but the perception of a face. What is the link between, so you've seen mid FDs having an additional sort of being extra good at triggering the perception of a face in a human. What is the relationship, do you think, between that magic number or that magic range of FD and actual human faces? Because we've clearly evolved to have this ability to see faces really quickly. What does that say about us and faces?
First of all, I need to just clarify that, like, during the task it was not specified to participant to search for faces. So sometimes it was like, I asked like, I have a little bit of qualitative data, so it's mostly faces, but sometimes it's like mythological creatures or whatever. But to answer your question about like the 1.3 number, I think it mostly relates to what we are used to find in nature. So fractal dimension around 1.3 is kind of really natural. There has been some studies that they do in virtual reality. You have like structures that you need to navigate in, and if you have fractal dimension 1.3 in those structures, you can easily navigate and remember where you were. But if you have more complex structures, you get lost. And if you have less complex structure, then you don't know where you are because you don't have like any place to remember, you know. So I think it really relates with what we are used to see. So in nature, you can find like 1.3 pretty everywhere. So I think this is why like even low creative individuals will mostly rely on fractal dimension 1.3, but high creatives, because I think they rely on pareidolia more often. This is maybe why we show that that they are able to experience pareidolia in a wider range of fractal dimensions.
Right, awesome, thank you. Back to Chiara with a question from Matthias Brucklacher. I'm sorry, I totally butchered that. I apologize. How is a memory defined in your network? Is it a static or a dynamic activity pattern?
Okay, so first we train our network over a certain number of examples, and then we store each example in the synaptic weight matrix. So each example becomes a memory, and it is at the beginning stored in a static way into the synaptic matrix. Then during the sleeping phase, the weights of these synapses are plastic, so they can change during the time. And this means that the representation of these memories, so these memories are actually changing during the sleeping time. And that's depicted in the images I showed you, where blue and red points moved in the space, just grouping together. So I would say that's a dynamic interpretation of that during the sleeping phase.
Right. Damn it! I want to ask my own question, but fine. I'll go with Jennifer's question here. So Chiara, still, would you speak more about your energy and entropy measures and how you might interpret them with respect to biology?
Okay, from the energy measurements, it's quite easy. We compute the energy according to the neural activity, so firing rates of neurons weighted on the synaptic weights connections. So higher activity, higher energy consumptions, higher weights of synapses, higher energy consumptions, and so on. And that's quite relatable to biological measurements. On the other hand, the entropic information, it's quite hard because it relates to the interpretation of the firing rates of different parts of the cortex, so different groups of cortical neurons. And that's something we'd like to discuss a little bit more with you in order to get an idea of how, again, to anchor these theoretical measurements we are trying to propose and some biological experiments. And also, if you have any idea, please email us because we will be happy to discuss that with you.
I have a strong feeling Jennifer Goldman will be in touch because this is something she works on. Jennifer, if you feel like adding anything to this, you can raise your hand and we can put you live on air. So thank you, Chiara. Until then, Antoine, is scale important in the fractal dimension you computed for each stimuli?
I'm not sure exactly I get what you mean by scale, but like if when we generate fractal images, there is that 1/f bound. So the idea is that by varying that, so sorry, I just have the name in French, that slope, by varying that slope, you can vary the fractal dimension of the image, but the idea to have more complex images, it's basically noise that is built in with different octaves, and the more octaves you add, the more complexity you will have. So it's like, it's the idea of, it's similar to Perlin noise. Perlin noise has been used in like all generative models used in like video games and things like that to generate textures. They are all based in like fractal noise, and these are the idea of generating noise at different scales, and you build them together, and the more octaves you have, the more complexity you will get in your noise, basically. And then we computed the fractal dimension on the images afterwards to validate that these images were at the good fractal dimension. So I'm not sure if this answered the question.
If it doesn't, they know how to get back to us. I was also thinking whether you were considering using a different technique for generating your stimulus, maybe using a generative adversarial network or something. You could at that point train a GAN to essentially exploit the pareidolia in humans to really force them to see patterns.
This is a great idea, because one thing we were thinking is like with all those deep dream things, like there is the idea of like comparing human and machine pareidolia, because like deep dream is mostly like a machine pareidolia somehow. So we are starting to think how to use machine learning to generate stimuli to increase pareidolia, and also the idea will have to build like, because I'm working also on brain-computer interfaces. So the idea will have to have like real-time loop to increase pareidolia in real time. So if we can decode in the brain what is happening during pareidolia, and we can translate this into a feedback, reinforce pareidolia, then we can have something really interesting. But it's not my main expertise, but the lab I'm working in, which is at University of Montreal, is mostly focusing on computational neuroscience and machine learning. So these are in the next steps for sure.
That sounds very interesting, and I hope someone in the chat raises the ethical questions of hacking a human mind to enforce a certain perception on it. I'm not going to be the one raising that. So, Chiara, this person loved your presentation and was very interesting. Could you please elaborate what age group and how many hours of sleep was considered during the experiment? So could there be a correlate between their small modeled network and the life of a...
Yep. Okay, I need to clarify that this is a simulated model. So no age group, because it's almost my computer. Still, our sleeping simulation is about 20-minute simulation. It emulates kind of just one step of deep sleep-like activity. And this is quite coherent with what we are trying to perform, because it is almost a simple task. So we, reasonably don't need the whole night of sleep, but just a nap of 20 minutes will be good enough.
Awesome.
Yeah.
Okay. What kind of simulator do you use for this? Because some of the stuff that you described with the cos theta measurement sounds a bit like what Nango, the Nango simulator, is trying to do with their synaptic pointer architecture, if I'm remembering correctly, and their concepts and distances between concepts.
The rate model is done. We just coded in Python, and it was just built within the laboratory, whereas the spiking simulation is performed by Nest.
Okay. Thank you. So going back to Antoine, did you say that a particular type of entropy is associated with creativity? Do you think such entropy could support creativity, and how? You're muted.
Sorry. So this is a good question. What I've been talking about was one recent study that shows relation between brain entropy and divergent thinking, but that was fMRI study. So I'm not an expert in fMRI, so I think it was kind of a classical measure of entropy that they use. I know there is also those multi-scale entropy measure that are been using with EEG, because I'm more focused on EEG and MEG signal. So there are hypotheses relating to the idea that maybe if we have a more complex stimulation, maybe the pareidolia during the perception of those kind of stimulation will increase entropy at the same time. And also there is some theories of creativity that kind of conceptually relate to the idea of entropy, because we have that like two phase in creativity. You have that exploration phase and then that validation phase. So during the exploration phase, for sure, having higher entropy, higher information is really coherent with the idea of being able to have remote association and to make links between unrelated concepts. So to me, there is clearly a relation between entropy and creativity. And also there is that entropic brain theory from Harris trying to explain the effect of psychedelics. We know that psychedelics and creativity are pretty related. So this is another study that goes in the same line for relating entropy and creativity.
If I'm not mistaken, sleep is also very related to creativity. Have you had the chat with Chiara?
Not yet.
Okay, it sounds like you should have a chat. So going back to Chiara, what is the biological intuition? Matthias asks again, what is the biological intuition for the synaptic weight matrix update rule during sleep?
Okay, during the training phase, we have a specific activation pattern in the thalamus for each example, and then we activate through a contextual signal also specific subgroup of cortical neurons. During the sleeping phase, a circular effect appears. So specifically, a specific subgroup of cortical neurons performs an upstate, and this will activate the pattern associated to the example over which it has been trained in turn. Then this activation pattern in the thalamus will activate other cortical neurons that have been trained over different yet similar examples. So this will lead to an enchantment in synaptic connections, and specifically the pattern describing each memories will be mixed together, and memories will be grouped in the internal representation. So the idea from a biological point of view is that looking at the activity of cortical neurons after the sleeping phase, you can see that if at the beginning all cortical neurons would activate in an orthogonal way, so only once a time, at the end of the sleeping phase, we will have that cortical neurons trained over similar examples, yet not the same example, will activate together, firing all together at a lower firing rate. So a concept will be created in the network, and not only example specific, but a more general concept will be created. I hope I answered the question. Not sure about it.
Once again, if you haven't, there's, I think we're now running out of time. So this is, if it hasn't been answered fully, then I recommend we go to Slack for that, including for the last question that I don't think we have time to answer now, which is, so Chiara, if you want to just answer it in Slack, that would be great. The question is, how do you perceive scaling up the network? So how do you see making this network larger? So once again, thank you everyone for joining. Thank you to our speakers, Chiara and Antoine, for gracing us with their lovely presentation and research today. The next HBP Tea Slides is going to be on the 8th of April at 2 p.m. So the same time slot as just now. And next month, the topics are going to be modeling the cerebellum and looking at the study of cortical lesions. We're still looking for speakers for the following months. So if you're an HBP student, please do volunteer. These sessions are tons of fun, I believe. If they're not tons of fun, email me and tell me that they're not. After the webinar is done, you will be greeted with a survey provided by Zoom. Could you please fill that in so that we get a better idea of what's working and what's not, so that we offer you the maximum knowledge transfer and enjoyment possible over Zoom. And if you have any more questions or would like to get in contact with the organizing committee or the speakers, more importantly, please look at the Slack channel organized by the HBP. And with that, thank you very much and see you next time. Bye.
Bye, thank you.
Thank you.